Okay, so today we will look into muscle biochemistry. So it's a bit of physiology of the muscle and also what are the roles of each component of biomolecules when muscle contraction and also relaxation occur. Okay, so that's generally about muscle biochemistry. Okay, so we will look into the classification of muscles. What are the types of muscles that we have in our body, and what are the components that makes up the muscle, especially the skeletal muscle. Okay, because we will look into the mechanism of contraction which occurs within the skeletal muscle, or what we call as the contractile machinery, and also the biochemical events during muscle contractions, which actually we will look into the role of calcium ion and also ATP. All right. So, okay. So in general, the muscle tissue in our body is responsible for the movement inside our body, of our body. So it's also responsible for the changes in size and shape of our internal organs. Okay, that's why we have different types of muscle tissues, which are skeletal muscle or the striated muscle, cardiac muscle, and also smooth muscle. Okay, so these are the examples in terms of the histological perspective of it. Meaning that, for example, over here we have smooth muscle tissue. So this is the morphology of it. And then we have cardiac muscle tissue and also skeletal muscle tissue. So apart from the differences in terms of their histological perspective, there are also difference in terms of their what activates their or what regulates their action. Okay, we can actually observe them or classify them based on voluntary or involuntary action. Okay. All right. So for the muscle, for the skeletal muscle, generally it connects the various parts of the skeleton through one or more connective tissue tendons. So we have our bones. So and attached to it the skeletal muscle. So during muscle contraction, skeletal muscle will shorten, and the shortening of this skeletal muscle actually moves the various parts of the skeleton. Okay. These skeletal muscles are activated through signals carried to the muscles via nerves. Or so these actions are voluntary, meaning it's on purpose. So when you want to hold on something or you want to walk, for example, so it's generally on purpose. So that's what we call as the voluntary control. It occurred, and this shortening or contraction of skeletal muscle, if it happens repeatedly, it can cause or lead to fatigue. Okay, at one time we feel fatigue. Okay, and there's this field of biomechanics which actually assess the movement and also the sequential pattern of muscle activation that move body segments. Okay, so these are the morphology of skeletal muscle. Okay, so it's surrounded by capillaries. So you can see capillaries. There's also adipocytes or the fat tissues. Okay, there's also sarcolemma, one of the layers that or the sheath that actually covering the skeletal muscle. Okay, so another types of muscle is smooth muscle. So smooth muscles are located in the blood vessels, respiratory tract, even the iris of our eye, and also the gastrointestinal tract. So compared to skeletal muscle, smooth muscle contractions are rather slow and also uniform, and they function to alter the activity of various body parts to meet the needs of our body at that time. And generally, smooth muscles are fatigue resistant, meaning they hardly become fatigued, although they are repeatedly activated. And the activation of smooth muscle is rather involuntary. So this is important for us to understand. For example, if the smooth muscle inside our respiratory tract are prone to fatigue, for example. So at one time, after breathing for a few hours, the muscles may get fatigued and it will stop functioning. So it's very dangerous. So that's why smooth muscles are involuntary and also fatigue resistant. Okay, in terms of the activation. Okay. So the next type of muscle would be cardiac muscle. So where is this cardiac muscle? Where can it be found? The heart, yes. Cardiac muscle or the heart muscle. So this cardiac muscle, it has the characteristics of both skeleton and also smooth muscle. Okay, so it functions to provide the contractile activity of the heart, and the contractile activity can be gradated, and but they are very fatigue resistant. Okay, and the activation of cardiac muscle is involuntary. Okay, meaning that we don't have to instruct our hearts to start beating, right? So that's why it's involuntary. Okay, so that's generally about the types of the classification of muscles. Okay, let's look into the components of skeletal muscle for us to understand how the physiology of muscles, all right? So if we zoom in, this is the muscles within our hands, all right? So this is the tendon that actually connects our muscle with our bone. Okay, so within this area, this cross-section area, these are the muscle bundle. Okay, this whole thing over here is the muscle bundle. Let me annotate this. Okay, this is muscle bundle, and each muscle bundle are made up of a lot of fascicles. Okay, so if we zoom in, this one part over here is muscle fascicle. So muscle fascicles are made up of a lot of muscle fibers, right? So within these muscle fascicles are a lot of muscle fibers. So if we zoom in further, these muscle fibers, we can observe this cross-section of myofibril. Okay, so these are, if we further zoom in, these are one unit of sarcomere, which makes up this area over here. Okay, we will look into the details later. So this is just actually the anatomy of our muscles, right? Okay, so over here we have the sarcomere, one unit. So this one whole area is one unit of sarcomere. Okay, so this is the details of sarcomere. So sarcomere generally are made up of myosin and also actin, the thick filament and also the thin filament. Okay, so this is the illustration of it. One unit of sarcomere contains actin filament. So actin filament is the blue colored line over here. Okay, the filament, the blue colored line. So this is actin filament. So another important unit would be this myosin filament or the thick filament. Okay, the yellow colored ones. So these are tiny like projections over here are the myosin head. Okay, we will look into that in detail later. Alright, so this area from one end of myosin filament to another end over here. Let me annotate this. Alright, so this one end to this end is what we call as the A band. Alright, and then we also have I band, which is the end of this myosin filament within this sarcomere unit to the next sarcomere unit is what we call as I band. Alright, we also have the Z line over here, okay, which holds the actin filament, the Z line. Okay, and then we have M line. Okay, the center in between the myosin filament, the area which doesn't have myosin head. Okay, okay. Alright, so that generally or briefly are the components of skeletal muscle. So let's look how our muscle contract and relax, or the contractile machinery. Okay, so the theory of how our muscle contract or move is based on this theory of sliding filament. Okay, so these are based on these two action of thick filaments, which are the myosin. Myosin would be these yellow colored ones, and also the thin filaments, which is actin, the blue colored filament over here. Alright, so these two plays a role in representing how the muscle actually contract and relax in this sliding filament model. Right, so this is again the image of sarcomere and also the details of it. Right, so this sarcomere is actually the entire contractile unit, okay, from this Z line to this Z line, which consists of actin filament, myosin filament, and also these bands and also lines. All right. Okay. So how does what triggers the contraction of muscles is basically based on the activation from nerve. So action potential will arrive at the neuromuscular junction. Okay, I will just go through this briefly because this is physiology, right? So this is the neuromuscular junction. Neuromuscular means it's where the junction where neurons and muscle meets. Okay, so the action potential will arrive at this neuromuscular junction. Okay, action potential arrive over here. Okay, so what's next? So when action potential arrive at this junction, it will trigger the release of acetylcholine. So within these vesicles are actually acetylcholine, or the short form would be ACH. Okay, so this is an important neurotransmitter, especially in muscle contraction. Okay, so the release of acetylcholine over here will activate the sodium channels, sodium ion channels. So these, the purple channels over here are sodium channels. So activation of these channels will cause the influx of sodium into sarcolemma. So meaning that after activation, so now this channel will allow sodium ion to move into sarcolemma. Right? So this area is the sarcolemma. Okay. And the influx of sodium will trigger action potential. Sorry, this is not actin. Action potential in sarcolemma. Okay, so sodium influx. So now there's action potential propagation, point number 3. Okay, and this action potential will propagate through this T tubule over here. Alright, so when it reach this area, sarcoplasmic reticulum, it will trigger the release of calcium ion into the cytosol. Okay, so now the propagation of action potential will now causes the influx of calcium ion into the cytosol. Right, so calcium ion will move in. So what will this calcium ion do? It will bind to troponin. Okay, so later on we will look into the detail which one is actually troponin, tropomyosin, and myosin head. But just to give you an idea of how actually muscle contraction starts, what triggers it. Alright, so the important point here is basically it must start with this action potential arriving the neuromuscular junction, and then release of acetylcholine, propagation of sodium ion, and then triggers release of calcium. So this calcium will go into the cytosol and binds to troponin. Alright, so up until point number 5 is, oh my god. So up until point number 5 is actually the initial starts of muscle contraction, and point number 6 onwards we will look into detail in the next slides. All right. Okay, so to give you an overview of what sliding filament theory means, so this is the diagram or the illustration of when our muscles are relaxed. Okay, so everything has distance, okay, in between them. So when the muscle contracts, the actin filaments will slide into the A band, which will cause them to be overlapping with myosin. Okay, so if you notice over here, upon muscle contraction, so actin filaments will slide. Okay, this area will slide into the A band. Okay, it will move in that direction. Okay, it will contract like this, right? So what happened is that the Z line will move closer together. So if you notice, these Z lines over here will move closer to each other. Okay, it will become shorter like that. Then the I band, what happens to the I band? It becomes shorter and the A band stays at the same length. Okay, because the myosin filament will not change in terms of its length. Okay, only the actin will be moving. Okay. So this is the overview of it. Okay, so what happens, what causes this actin filament over here to move, okay, and become eventually overlap with the myosin is that, okay, so because of the myosin is actually attaching to the actin and pulling it. Okay, so this is, if you zoom in a bit more, so this is the myosin filament. So these tiny projections are the myosin head. Okay, so this area is what represents this area just now. Okay, so the filament will slide together because myosin attaches to actin and pulls it. Okay, so this myosin head, this is the myosin head. Okay, attaches to actin. So this is the actin filament, the yellow colored one, and forming cross bridge. Okay, when they are attached, we call it that condition to be in a condition of cross bridge. So this, after the cross bridge is formed, the myosin head will bend and pull the actin filament and causing them to slide. Okay, so if you want to imagine this, muscle contraction is like climbing a rope. Okay, so we attach and then pull, attach and then pull. Okay, right. So let's look into next. Okay, so these are the biochemical event during muscle contraction. So it will start with the binding of ATP. So there's involvement of ATP, which means that muscle contraction requires energy, okay, to the head of myosin. And then we will observe upon binding of ATP, there will be conformational changes in the myosin head. And we will also observe the binding of myosin to actin filament, okay. And then there will be a release. Myosin head will be released to allow relax. So the contraction will not be for too long. Okay, it will be contract, relax, all right. And there's a reattachment of the myosin head, and the cycle will continue. All right. So this is the diagram for it. We will look into one by one. Okay, before that, let me show you first the video for this one. That's here. Okay, so this is the neuromuscular junction. Okay, so action potential arrive at the neuromuscular junction. And now, okay, so this is acetylcholine. So acetylcholine will bind and activates the sodium channel. This is sorry everyone. Continue with the connection. So, sorry. So when calcium ion moves into the cytosol, it will bind to the troponin. Okay, upon binding of calcium, this is what happens. It will turn proactive filament. Okay, this is the tropomyosin. Right. So this is tropomyosin. Within the actin filament, the dark green circle over here is the binding site for this myosin. So this purple here is actually myosin. So this is the thick filament just now. This is the actin. Okay, so it has the binding site for myosin. So
Dokter, suara dokter tak dengar.
yes, now better?
Yes, better.
Okay.
Dokter dah record.
Dah. Dia tak stop pun. Pause on tadi. Okay. So again, calcium ion will move into the cytosol and it will bind to this troponin, the orange color one. okay, and cause the conformational change of troponin and also tropomyosin. So they will turn just now as you observe, and it will cause this binding site to be exposed to myosin. Okay, so it's easier for now the myosin head to bind to this actin. Okay. Okay, so concurrently, what will happen is that ATP will bind to myosin head, and the hydrolysis of ATP will control the action or the binding of myosin head, okay, to the actin. So now it will move closer to each other. Okay, so calcium goes in, causes the conformational change of the actin filament, exposed binding site to myosin head bind because of the ATP that is attached to it. So after ATP has been hydrolyzed, myosin head will detach from actin. Okay, and the cycle repeats. Okay, and just like that. Okay, then move and then slide. So that's how actin is getting closer to each other like that. Okay, so this is this very slow motion version of it. Okay, so in normal or under normal physiology, things would happen very fast. That's why we are able to control our skeletal muscle. Okay. Okay, so let's go into detail of it, the biochemical events of this, which we will focus on the calcium ion and also the changes that happens to ATP molecules. Okay, so in stage 1, so before this image diagram of A over here, what will happen is that ATP molecules will bind to the myosin head. Okay, so before this, before the hydrolysis into ADP and P, actually ATP will come in and binds to this myosin head over here. So this is the myosin filament, this is the myosin head. So binding of ATP will dissociate the ectomyosin into actin and myosin. So ATP binding will prevent the binding of myosin head to actin. Okay, so it induces conformational changes in the actin binding site of the myosin head and reduce the affinity of myosin head to actin. Okay, so generally means ATP, in the presence of ATP binding to myosin head, so it will prevent the myosin head from binding to the active site within the actin filament. Okay, so detached. So there's no cross bridge form over here. Alright. Okay. Now the time left is 6 minutes. Let's take 1 minute break and come back into the link. Okay.
Okay, doctor.
Yeah, doctor.
Doctor. Oh, sorry. Later you will give this slide in GC, right?
Yes, yes.
Okay, thank you.
Alright. Okay, so let's continue. So after ATP has been bind to myosin head, so stage 2 will happen. So what happened in stage 2 is that ATPase, the enzyme, will hydrolyze ATP into ADP and also a phosphate group, okay, which will remain bound to the myosin head. Okay, so after hydrolysis of ATP, ADP and phosphate group will still bind to the myosin head. So the energy, E is energy, okay? Energy release is stored within this myosin head, okay? So now the myosin, ADP and P complex, okay? All this bound together is called a complex, is now in a high energy state, right? So this is predominant state at rest, okay? So this is, okay, so ATP bind and then hydrolyze to ADP and P. So at this stage, it will start to bind to actin filament. Okay. So in stage 3, what will happen? So upon muscle stimulation, meaning that stimulation means when there's action potential from the neuromuscular junction. So the inhibition of actin-myosin interaction is lifted, and now the myosin ADP complex will bind to actin again, and actin-myosin reassociation triggers release of this phosphate group from the myosin head. Okay, so when myosin head attached to actin filament, and there's movement of myosin head, so ADP and also phosphate group will be released from the myosin head. Okay, so the myosin head is get tightly bound, okay, with the release of ADP to a new actin molecule. Okay, so the actin myosin complex will remain intact until another ATP molecule will come and bind to the myosin head. Okay, so it will start with this cycle back, stage 1 again. Okay, after stage 5, then repeat with stage 1. Okay, so until the movement of actin is significant, actin filament. Okay. All right. Okay, so again, in muscle contraction, we observe the importance or the significance of calcium ion. So without calcium ion, this troponin will not be undergoing conformational changes, allowing the myosin head to bind to the actin filament. All right. So influx of calcium is the trigger for muscle contraction. So in the resting state, protein tropomyosin winds around actin and covers the myosin binding site. Okay, so it will prevent the binding of myosin. But as calcium ion binds to the second protein troponin, this action causes the tropomyosin to be poked. This is what the conformational change that I've mentioned, and exposing the myosin binding sites. Okay, so when with the sites exposed, so muscle will contract if ATP is present. So calcium will prepare our muscle to be able to contract, but the presence of ATP will allow the contraction to occur. Right? Okay, so this is again the same machinery or mechanism, just that this is with the point of view of calcium ion. Okay, so in a resting muscle fiber, concentration of calcium ion is very low. Okay, and tropomyosin is located close to the myosin binding sites on the thin filament. So in this position, the tropomyosin blocks the myosin head from binding to actin filament, hence it's preventing contraction. Okay, but in a stimulated muscle fiber, the calcium ion released by the sarcoplasmic reticulum earlier will bind to the troponin. Okay, the calcium troponin complex will pull the tropomyosin away from the myosin binding site, so allowing cross bridge to form. So cross bridge is when myosin head binds to actin. Okay, so this cross bridge cycle continue as long as the calcium ion remains attached to troponin. So when nerve activity ceases, meaning that when the muscle stops receiving action potential from the neuron at the neuromuscular junction, okay, so impulses in the muscle fiber will also cease, and calcium is actively transported from the cytoplasm back into the sarcoplasmic reticulum. So within the cytosol, so now there's efflux of calcium into sarcoplasmic reticulum. Okay, so as calcium ion is released from the troponin, so tropomyosin will return to its inhibitory position, so it will prevent the myosin head from binding with actin. Okay. So this is important for us to note. So muscle contraction will stop when calcium ion is removed from that environment, meaning that from the cytosol back into the sarcoplasmic reticulum. Okay, and this action of pumping calcium ion back to the sarcoplasmic reticulum requires ATP as well. Okay, so the tropomyosin will reassume their inhibitory position back. So myosin cannot bind to actin. Okay, so this is the illustration of how calcium is pumped back into the sarcoplasmic reticulum. Okay, so in the point of view of the role of ATP, so when a skeletal muscle is at rest, the myosin heads function as enzyme, which previously I've mentioned ATPase. So this ATPase will cleave ATP into ADP and also a free phosphate group. So the hydrolysis of ATP will activate myosin head, okay, putting them in an orientation that allows them to bind to the specific sites of on actin over here. Okay, so it's about two states over here. The conformational changes that occurs within the actin filament itself. Okay, just now when we observe the role of calcium ion, so when we observe the role of ATP, it's about the conformational changes that occurs within the myosin filament, specifically within this myosin head. Okay. Okay, so that's why again ATP is required for the contraction of muscle. Sorry, someone asking something.
Doctor,
Who's
the
asking?
calcium
Making me
ions will be bind to the troponin and this apa ni concurrently with the presence of ATP.
Yes. So all of this happens at the same time. So it's not who
Ah,
comes first. Okay. So this happens at the same time and it's actually very rapid. Okay. So the purpose of this flow of lecture is for us to understand the contractile machinery, but in normal physiology, all of this happens rapidly and concurrently. Okay. So and it's
right.
actually a cycle. So we can't really tell who comes first. Okay, because it's ongoing. Okay, so again, so ATP is required for the sliding filaments to occur. It's also required for the separation of actin and myosin so that our muscle is not in a fatigue condition. Okay, when ATP is bind, remember, so it will cause the detachment of myosin head to actin. So it will prevent the muscle from becoming in a contracted state for a long time. Okay, so ATP allows the movement, but it also allows the relaxation to occur when it detaches the myosin head from the actin filament. Okay, so that's why in death, so ATP will runs down after death. Muscle goes into a state of rigor mortis. This is caused by the chemical changes in the muscles after death, causing the limbs to become stiff, okay, and difficult to move. Right. Okay, so that is about muscle biochemistry. So generally is for you to understand the role of calcium. So how calcium actually influence muscle contraction. Does calcium causes the contraction or prevent contraction? So if it causes contraction, how does calcium ion causes contraction? To who it bind? It will bind. And what conformational changes will calcium cause to this sarcomere unit just now? Similarly to ATP, so what is the role of ATP in muscle contraction? To who ATP will bind? And what conformational changes will ATP causes to this, for example, myosin? So that's briefly what you need to know or understand or even remember for this muscle biochemistry. Okay. So that's why in people who have calcium ion imbalance in their body, it may affect their movement and even sometimes, so this one is in skeletal muscle, so calcium ions are also important in smooth muscle, cardiac muscle, for example. So imbalance of this calcium ion and also ATP that is present in our body can actually affect the contractile machinery of our muscles. Okay, so it will affect the physiology of the organs itself. So we may have troubles in movement, for example, and some people may have bradycardia in the state of slow contraction of their heart muscles, for example. Okay, and some people may have conditions like constipation, so it's hard for them to the gastrointestinal lining doesn't contract accordingly, for example. Okay, so that's why in diseases that affecting the levels of calcium ion and ATP will causes troubles in, for example, movement. Okay. Okay, any question? Any parts that you need me to re-explain?
Doctor,
Hmm. Let me share.
so Doctor,
Hmm.
so the myosin will bind to the actin whenever the myosin get the ATP, and then the ATP will be hydrolyzed into ADP plus inorganic phosphate.
Okay, it's like this. When ATP binds to the myosin head will be detached from the actin. Only when the ATP is hydrolyzed into ADP and phosphate group is when this myosin head will bind to the actin filament. Okay, so all of this happen when these complexes are still attached together, meaning that after the hydrolysis of ATP, ADP and phosphate group will still bind to the myosin head. So that complex of myosin head, ADP and phosphate group causes this myosin head to bind to actin. So when ADP and phosphate group are released from the myosin head, so now there's movement sliding of the myosin head and at the actin filament, and another ATP molecule will come and bind back to the myosin head and cause the detachment of myosin head from actin. Okay.
Okay, Doctor.
Okay. Any more question? Okay, let me share again this video for you to get a better idea.
Muscle contraction. In this module, you will learn about the mechanism of muscle contraction, walking and running, that myofibril movements. The sliding filament theory is the most accepted theory which best explains how these proteins work to cause contraction of the muscles. The theory states that contraction of a muscle takes place by the sliding of the thin filaments over the thick filaments. Let us understand the steps given in the theory. The process of muscle contraction begins by a signal from the central nervous system, CNS. The signal is brought by the motor neurons from the CNS. These motor neurons are connected to the muscle fibers. The motor neuron with which a muscle fiber connects is called a motor unit. The junction between the motor neuron and the sarcolemma of the muscle fiber is known as the neuromuscular junction or the motor end plate. This junction is actually a synapse, that is a small gap between the neuron and the muscle cell. The signals from the CNS are transported as electrical impulses, also known as action potential, across the synapse. A neural signal, on reaching the neuromuscular junction, releases a neurotransmitter, acetylcholine, thus generating an action potential in the sarcolemma. This action potential spreads through the muscle fiber and causes the release of calcium ions into the sarcoplasm from the sarcoplasmic reticulum. This in turn increases the level of calcium ions in the sarcoplasm. The increased level of calcium ions leads to the binding of calcium with the subunit of troponin on the actin filaments. This interaction triggers a conformational change in the tropomyosin, thus preventing it from masking the active sites for myosin present on the actin filaments, leaving them exposed. With the active sites of actin now exposed, the myosin heads start a binding process with actin, thus forming a cross bridge. The energy needed for the cross bridge formation comes from the hydrolysis of ATP attached to the myosin into ADP and inorganic phosphate. The attached actin filaments are therefore pulled towards the center of the A band, bringing the Z lines closer to each other. This ultimately results in the shortening of the sarcomere, that is basically the contraction of the muscle. You can clearly see that there is a reduction of I band during contraction, while the A band remains of the same length. ADP and inorganic phosphate are released during the shortening, thus binding the myosin head to the actin tightly. The myosin remains attached to the actin until a new ATP molecule binds to the myosin head and the cross bridge is broken. The ATP is again hydrolyzed by the ATPase found on the myosin head, and the cycle of the cross bridge formation and breakage is repeated, causing further sliding. The cross bridge cycling sequence continues to repeat till the muscle fiber is excited by the signal from the CNS. As the signal stops, no further release of calcium ions from the sarcoplasmic reticulum occurs. This enables the pumping back of calcium ions from the sarcoplasm back into the sarcoplasmic reticulum. This pumping back of calcium ions results in the masking of active sites on the actin filaments. This in turn causes the return of Z lines back to their original position, that is, relaxation. All the muscle fibers do not respond to the signal in the same interval of time. That is, the reaction time. The interval between the signal and the beginning of a response is not constant in all the muscle fibers.
Okay, so that is just what you need to know for muscle biochemistry up until the role of calcium ion and also ATP. Okay, so any question? For muscle biochemistry, can you try on the tutorial question later? So the tutorial that I have given you last week, one question is with regards to muscle biochemistry. Okay, so please try and we will discuss them this week, Friday.
Okay, doctor.
Alright, doctor.
Yeah, doctor.